Hi, this is Dr. Kat again from Central New Mexico Community College. In this short video, we'll discuss the pancreas. And so this is video L. The pancreas is this very long, tapered gland that sits retroperitoneally. It sits behind the stomach, and it's a rather delicate gland. On a cadaver, it always falls apart very easily. Remember, it has that major duct called the pancreatic duct, which, by the way, actually also can split off to have its own entrance into the small intestine. But we see the major duct merging with the common bile duct to eventually form the sphincter of Oddi. The portion of the pancreas that sits the most or the closest to the small intestine we refer to as the head of the pancreas, and the more tapered end we refer to as the tail of the pancreas. The pancreas is divided up again into lobules, and we've enlarged one of those lobules. And then we see two major cell types. We're going to see cells that form what we call the acini, which are these little circular structures. They're essentially forming little glands, and the cells, the acinar cells, can then secrete their secretions that eventually are going to end up forming bigger and bigger ducts. We have a second group of cells that form what we call the pancreatic islets. Often you'll hear this referred to, or these islets are often referred to as the islets of Langerhans. All these terms that we give to these various structures when they still have somebody's name in it are these days considered as older terms, but they're still very much used by most anatomists. So what is the difference between the cells that make up the acini and the cells that make up the pancreatic islets? We're going to start with the pancreatic islets. They do not have a direct relationship to the digestive system. The cells in the pancreatic islets are producing glucagon and insulin. So they're basically the endocrine part of our pancreas. On the other hand, are acinar cells that form the acini, which then ultimately form the little ducts, which become the bigger ducts, and eventually the pancreatic duct. They form the pancreatic juice, which contains a watery bicarbonate solution as well as many enzymes. Let's first focus on that watery bicarbonate-rich juice that the pancreas produces, which, by the way, has some electrolytes in it as well. Because it is rich in bicarbonate ions, it can actually help neutralize the chyme that is very acidic that arrives from the stomach into the duodenum. Because it has a high pH, this pancreatic juice, this bicarbonate-rich juice, it's an optimal environment for many of the brush border enzymes of the small intestine, as well as the pancreatic enzymes, which we'll take a look at in just a moment. Now, where do these bicarbonate ions come from to help form this pancreatic juice? Well, it comes from the blood. And by making bicarbonate ions, remember, we need to, let's review this real quick, we need to combine water with carbon dioxide. That then forms our carbonic acid. And then the carbonic acid can fall apart into hydrogen ions and bicarbonate ions. So we're left behind in the blood with hydrogen ions if we remove the bicarbonate ions to make our pancreatic juice. Consequently, the blood that leaves the pancreas has quite a few hydrogen ions in it, and you know what that does to the pH. It lowers the pH of the blood. This is okay because if this more acidic blood now mixes with the blood that leaves the stomach, remember we talked about the alkaline tide of the stomach, we see that the blood gets neutralized. The pancreas produces a whole slew of enzymes, and we're just going to list some of the more important ones. But because the pancreas produces so many important enzymes to help digest our nutrients, it explains why pancreatitis and especially pancreatic cancer can be so detrimental to a person's health. First of all, we find that the pancreas produces all kinds of protein digesting enzymes, better referred to as proteolytic. Remember what the word proteolytic means: proteolysis, the lysing of proteins. They're once again released in inactive form because otherwise they would start digesting the cells around them. But by the time they make it to the duodenum, they can be activated. So these are some of the three major proteolytic enzymes of the pancreas. Trypsinogen needs to be activated to become trypsin. Chymotrypsinogen becomes chymotrypsin when it's activated, and procarboxypeptidase turns into carboxypeptidase. And some of these proteins help each other to convert, kind of like a cascade effect, as we saw in the clotting factors. Some enzymes are secreted active, enzymes that help with the digestion of carbohydrates and lipids and nucleic acids. We can call them the lipases, the nucleases, and so once again we come across amylase. Recall that we found amylase in the saliva, so we have a form of salivary amylase that begins the digestion of complex carbohydrates, and here we see pancreatic amylase that continues the digestion of complex carbohydrates. Many of these enzymes require a variety of ions, electrolytes, or even bile in order to function most optimally. We see the same hormonal and neural mechanisms regulate the secretion of the pancreatic juices. So once again, we're looking at cholecystokinin and secretin. And both of these hormones are going to stimulate the pancreas to produce their enzymes and to produce their bicarbonate-rich juice. The vagus nerve has only a weak influence on the production of pancreatic juice, just like it had a weak influence on the gallbladder. This wraps up our discussion of the pancreas. As a matter of fact, it wraps up our discussion of the whole digestive tract and all of the accessory structures. So what is still left? Well, we still need to take a closer look at the chemical digestion of our nutrients and how they are absorbed. And so that's what the following few videos will cover.